Yeah, thank you, Peter, for the introduction. I'd also like to thank the organizers for giving me the opportunity to talk at this increasingly prestigious Cycle Club conference. So today I'm going to share with you our recent work in preparation about oncogene-induced whole genome doubling. So tumor cells are frequently aneuploid, as demonstrated by this graph, showing chromosome numbers in different tumor samples. This graph also shows a bimodal distribution, which represents two types of numerical aneuploidy. The first type shows minor gains or losses of chromosomes. It is caused by chromosome instability, which arises from errors in mitosis. The second type shows extensive aneuploidy, often containing a hypertriploid chromosome number, which can be attributed to a whole genome doubling event where every single chromosome is duplicated and then followed by chromosome loss. So this project was initiated when a former colleague, Stephanie Hills, was studying an oncogene called Cycle E. So Cycle E is a cyclin that regulates G1 S-phase transition and also initiation of dereplication. It is an oncogene that is overexpressed in many cancers. So we have an inducible system that can induce Cycle E expression upon treatment of doxycycline. And we and others have tested Cycle E expression in U2S cells, showing that Cycle E expression induces replicative stress. And interestingly, over time, we found that upon Cycle E expression, there is whole genome doubling happening, as shown here that about 6% of the cells are incorporating EDU with more than 4 NDN content. And the longer you overexpress Cycle E, the more whole genome double cells you will get. And we thought this is very interesting, and this is just the beginning of this project. We could show that these whole genome double cells are viable, and we were able to isolate these whole genome double cells by single cell sorting. We grow them into single cell clones and karyotyped the clones using metaphase spreading. What's quite interesting is that there is a wide spread of chromosomal numbers in cells within each clone. So you can see and most cells have chromosome numbers within each clone have less than the double of the original cells. So what you're looking here is similar to what is happening in cancer. So there is firstly a whole genome doubling and then followed by chromosome loss. So we thought this is very interesting, and this project aims to figure out how Cycle E overexpression leads to whole genome doubling. So to study this, we used a Fuchs live cell imaging system to realize the cell cycle. So the Fuchs system consists of fluorescently tagged CDT1 and germline fragments that are expressed in different stages of the cell cycle. In G1, there is only CDT, so the cells will look red. And in S phase, there are only germline, so the cells will look green. And in G2, both fragments exist and the cells will look yellowish. And for better cell tracking, we also added a fluorescently tagged H2B protein that is expressed in all the phases of the cell cycle. And we have also created an automated cell tracking system based on TrackMate. So first, I'd like to tell you that 3 possible routes to whole genome doubling. The first one is by cell fusion, where 2 cells fuse together to generate a binucleated cell. And the second is through failures of mitosis or cytokinesis. And depending on whether cells fail at the cytokinesis stage or before anaphase, it can generate either a binucleated cell or a cell with a foreign nucleus. The third route is through endoreduplication, where cells bypass mitosis, entering from a G2 directly to a G1 phase. And the difference between 2 and 3 is that there is no nuclear envelope breakdown or any signs of mitosis for mitotic bypass. So first, we want to figure out which of these 3 is happening in Cycle E expression cells that leads to a whole genome doubling. So we look at this by the Fuchs imaging technique I just mentioned. First, I will show you an example of a control cell. So the Fuchs channels are on the left, H2B channel on the right. So we can see the cells are going through all the phases of the cell cycle and is dividing. And we can track the cells based on the H2B channel and measure the intensities of the HCDT1 and germline future reporters and plot intensities against time. This allows us to directly label different phases on the graph. And the H2B channel is very useful, as in mitosis, the H2B signal spikes up, so we can indicate mitosis. We then look at the cell overexpressing cyclin E. So right now, this cell is yellow in G2, and it's supposed to have mitosis, but instead of having mitosis after that, it goes into a red G1 phase straight away. And on this intensity plot, you can see that the cell has a very long G2 phase, and followed by that, germline is slowly degraded with no signs of mitosis. And so indicating the cells has entered a G1 state that we call endured duplication cycle G1 phase, which I will call ECG1 for the rest of my talk. And we have evidence that the anaphase promoting complex is required to degrade the G2 cyclin cyclin B during this mitotic bypass process. And from many individual cells tracked by this system, we can quantify that about 30% of cells have bypassed mitosis in cyclin expression. We can also measure the length of the G2 of different cells. It shows that the cells that have bypassed mitosis in cyclin expression had significantly longer G2 than cells that did not bypass mitosis or in control cells. So these cells probably have suffered higher amounts of replicative stress compared to the others. So now we show that cyclin E expression leads to mitotic bypass, and then cells become whole genome doubled. And the obvious next question is that how cyclin E expression might lead to mitotic bypass. As I briefly mentioned earlier, the cyclin E expression causes replicative stress, as seen here in this quantification graph of the facts analysis on the left. There is an increase in gamma H2X in S phase in cells overexpressing cyclin E. And also cyclin E expression reduces the replicative helicase MCM from the chromatin. And this is because cyclin E expression accelerates and shortens the length of G1 so that cells don't have enough time to load MCM onto replication origins. So we show that cyclin E expression leads to mitotic bypass, and it also induces replicative stress. We then wonder whether there is a direct link between replicative stress and mitotic bypass. But then next, I'm going to show you that actually the kind of replicative stress is not important. So we generated cells inducibly expressing other oncogenes, CTC25A, CMYC, and oncogenic RAS. All of these oncogenes induced whole genome doubling to some degree. And interestingly, if we treat cells with N-acetylcysteine, which is a reducing agent, it can reduce the replicative stress caused by RAS overexpression, and also reduce whole genome doubling caused by RAS overexpression. This is consistent with studies showing that RAS generates reactive oxygen species to cause replicative stress. But in contrast, cyclin E expression was not affected by N-acetylcysteine, indicating that cyclin E expression generates different kinds of replicative stress from RAS expression. We also tested our cells with the phytidylcholine, which is a DNA polymerase inhibitor. It turns out this direct inhibition of replication can induce cells to bypass mitosis a lot, as in this future quantification, it shows that 80% of cells bypass the mitosis after long treatment with a phytidylcholine. And if we treat cells with the CHEK1 or WEE1 inhibitor while the cells were resting in G2 before they bypass mitosis, it can force them to enter mitosis. But these forced mitoses are mostly aberrant and generate daughter cells with aberrant nuclei. And a further look at facts on these ECG1 cells that bypass mitosis. At this point, at $70 treatment, most of the cells are already in ECG1. And we can show that these cells, they have loaded MCM onto the chromatin. And if we wash away acetylcholine and then release the cells into fresh media, the cells can further replicate, as shown here, that a massive 40% of cells are now having more than 4 ND in content. These results led us to think that actually replicative stress itself can induce mitotic bypass. And in fact, there has been a study about stress-induced endoreduplication in literature. So in this Davely and Delan 2010 paper, they show that mitotic bypass occurs following telomere attrition or double-strand breaks. But in their study, they require the absence of p53 for their cells to further endoreduplicate. So this is understandable, as p53 is a tumor suppressor that maintains genome integrity. So we also wanted to test whether p53 is important in our case. However, the p53 status of U2S cells that I just showed you can be very complicated due to MDM2 expression and RP16 deletion. So we want to use RP1 cells, which is a telomerase immortalized epithelial cell line, and is p53 plus. First, we just treated RP1 cells with the acetylcholine to see whether they bypass mitosis. So let me play the video. So you can see right now the cells are mostly in S or G2. But instead of going through mitosis, you can now see the cells turns red, which means they are now in G1. So these cell RP1 cells in acetylcholine, they also bypass mitosis and enter the endoreduplication cycle G1 phase. Then we wonder whether this RP1 EC G1 cells can further replicate. So we just wash away acetylcholine and release these cells into fresh media. However, it turns out these cells all stay arrested in ECG1. And unlike U2S cells, which I just showed you, these RP1 cells, they do not further replicate. And look at MCM loading shows there's no MCM loading in this RP1 ECG1 cells, indicating they are not entering the cell cycle again. So acetylcholine-induced replicative stress causes cells to to bypass mitosis and enter ECG1. For U2S cells, they can continue through the endoreduplication cell cycle, but for RP1 cells, somehow this cell cycle re-entry is suppressed. We wonder whether this suppression is mediated by p53. So to test this, we asked, can p53 knockout allow cell cycle re-entry in RP1 cells? So we just knocked out p53 in RP1 cells. And the first thing we tested is to treat these p53 knockout cells with acetylcholine. In wild-type cells, as I just showed you in the last video, they all bypass mitosis and enter ECG1 in acetylcholine. And we were expecting the same should happen to the p53 knockout cells. But let's take a look at the video. So very surprisingly, that the knockout cells behaved very differently from the wild-type cells, that you can see they still go into mitosis as you can see the cells have rounded up. And this quantification shows that mitotic bypass is not happening in these p53 knockout cells. So we were quite confused by this because in the Davely and Delan paper, they clearly show that p53 negative cells could bypass mitosis following telomere stress or telomere attrition. But in our case, unlike the wild-type cells, almost every single cell bypassed mitosis in acetylcholine. The p53 knockout cells, they just enter mitosis, and these mitoses are frequently catastrophic. They either end up with fragmented nuclei or they had mitotic death. And we also knock out p21, which is CDK inhibitor activated by p53, and found that p21 knockout cells behave like p53 knockout, and they also enter catastrophic mitosis in the acetylcholine. The same also happened when we overexpress cyclin E. So in wild-type cells, when they overexpress cyclin E, about 30% cells can bypass mitosis, but the p53 knockout cells, they do not bypass mitosis anymore. But instead, they had this catastrophic mitosis, and similarly as in acetylcholine treatment. And this is not because p53 knockout cells cannot arrest in G2 anymore. In fact, the p53 knockout cells still have quite long G2 arrest in either acetylcholine or second E expression. And they have longer G2 even than the wild type cells. So both p53 positive and negative cells in replicative stress, they could arrest in G2, whereas eventually the p53 positive cells, they bypass mitosis. The p53 negative cells, they have catastrophic mitosis. And these results led us to propose that p53 is specifically required for mitotic bypass. So acetylcholine-induced replicative stress activates p53 and p21, which then causes cells to bypass mitosis and enter ECG1. At this point, it's fine because p21 further inhibits, suppresses cell cycle re-entry of this ECG1 cells, doing its job as a tumor suppressor. Please don't feel confused that p53 and p21 is doing two things here. It is causing mitotic bypass and also suppressing the ECG1 cells. However, next, I'm going to show you that second E expression can potentially mess up with these tumor suppressors. As previous slide shows that RPE1 p53 wild type cells, they bypass mitosis in the acetylcholine, but they stay arrested in ECG1. However, with second E of expression, this is different. In second expression, the cells also bypass mitosis, but instead of arresting ECG1, these cells, we show they could continue through endoduplication, as in future live cell imaging. And also by fax, you can see these cells are now replicating, incorporating EDU with more than 4 DNA content. So we asked, can second expression overcome the ECG1 arrest? To test this, we first generated ECG1 cells by just treating RP1 cells with acetylcholine for 72 hours. At this point, most cells should have entered ECG1. And then if we wash away acetylcholine and release the cells into fresh media, the ECG1, RP1 ECG1 cells will just stay arrested in ECG1, and very few enter S or G2 or M phase. But if we induce second E expression at the point of release, this could drive the ECG1 cells into S phase, and the cells can continue through the cell cycle. And looking at FACS analysis, it shows that second E expression can promote MCM loading in those ECG1 cells. So to summarize, we show that persistent replicative stress, like in acetylcholine treatment, induces p53 and p21 activation. I don't have time to show all of the data, but we think p21 potentially inhibits CDK1, leading to activation of APC-CDH1, which in turn degrades cyclin B, thereby cells bypass mitosis and enter a ECG1 phase. Cells at this point are whole genome doubled, but it is not dangerous yet because p21 further suppresses the growth of the resulting ECG1 cells. However, when there is second E expression, it can not only induce all of this down the process, but it further promotes cell cycle re-entry of the ECG1 cells, enabling them to continue through the endoduplication cell cycle, undergo 8N mitosis, which we have results to show that is prone to chromosome segregation errors, potentially causing aneuploidy. But this is not the end of the talk yet. All of these I'm telling you is what we see in cells. In fact, our finding is supported by data in real tumors. So these recent large-scale genomic studies are looking at whole genome doubling in cancer patients. They reveal that whole genome doubling often follows a preceding driver mutation. And such mutations include overexpression of oncogenes like cyclin E. And cyclin E is able to drive, this can even happen in p53 positive tumors. So some take-home message for you, the replicative stress can lead to whole genome doubling by inducing mitotic bypass. And we show this mitotic bypass is dependent on p53 and p21 activation. And we show second E expression further promotes cell cycle re-entry of cells that have bypassed the mitosis, i.e. the ECG1 cells. So we propose that being the most likely early source of replicative stress, oncogene expression is a driver for whole genome doubling in cancer. So that's the end of my talk, but I'd like to thank our former colleague Steph for initiating and working with me on this project, and also ECHO in the lab for help with generating cell lines. I'd also like to thank everybody in the DFD lab, especially John, for help and support throughout my PhD, and also Rob, Simon, and Nicholas on my thesis committee for providing valuable feedback. I also thank John and Joe and Sylvia for help with making the future cells. Last but not least, I'd like to thank the Crick facilities for providing excellent technical support. So I'm ready to take questions from you.
Great, thank you very much, Jingkun. There were questions in the chat. I'll perhaps start with Sayak. Sayak, can you unmute and ask your question?
Yes, I was wondering, so what would be the fate of this p53 deficient cells which have undergone whole genome duplication? Would they die out of the population at some point, or they would add to the tumor volume, aggressiveness of the tumor?
Yeah, thank you for the question. Yeah, you mean the p53 negative cells or the p53 positive cells?
Negative cells.
The negative cells. We could show these cells are viable, but we didn't do a competition assay to say whether they die out from the original cells. But we did isolated clones from those whole genome duplicated cells, and we show that they had very number of chromosome numbers, if you remember in the first few slides. So yeah, they could continue proliferating, and we also show that those cells have increased chromosome segregation errors.
Thank you.
Yeah.
Okay, Brian. Question from Brian Calvi. Brian.
Yeah, thanks for the talk. My question's about your p53 inhibited RPE1 cells, and the surprising finding that you need p53 for whole genome doubling. Did I understand that correctly?
Yeah, it is required for bypass mitosis, yes.
Yeah, but there's a lot of other cell lines that are p53 compromised that don't have p53 activity that will do whole genome doubling. Yeah. When you stress them or do something to them. So I guess my question is the p53 requirement specific to the RPE1 cells then? Because it's not required in other cells for whole genome doubling.
We also tested a panel of other cells like HeLa cells and HCT116. We all show that if they don't have p53, they undergo catastrophic mitosis in acetylcholine. But I guess if you don't treat them with too much stress, they could maybe fail mitosis and become whole genome double. But that's through a different pathway. What we show here is these cells bypass mitosis. They don't become whole genome double through a failed mitosis or failed cytokinesis.
So it's the type of stress and the level of stress then is what you're saying. So if you do a lot of acetylcholine, they don't restrain mitosis, but then if you do a little, then they do restrain mitosis, then you don't need p53. Because it was interesting because p53 is involved in the tetraploid checkpoint too. So anyway, yeah thanks for the answer. It's complicated, but okay, thanks.
Question from Frank Springer. Frank, are you able to unmute?
Yes, hello. I'm just wondering, when you get overexpression of cyclin E, then you basically have stress, and the cells will end up in a G1, have high levels of p21, and then cyclin E can probably overwhelm the p21 inhibition. So you should get the same effect after p21 depletion. Have you tested that?
You mean like a p53 depletion?
Well, no, to get the same, so you take cyclin, you take a different stress. Yes, you take a different stress, and then get also genome duplication, and then you remove p21.
Yes, we have done that, and it is like so p21 is required for mitotic bypass, but then it suppresses the growth of the bypassed cells. So if you deplete p21 in those bypassed cells,
yes,
they can further replicate. It's like when it's like you induce cyclin E in those cells.
Okay.
So yeah.
All right. Thank you.
Okay. Thanks. A question from Joseph Forment. Joseph, can you unmute?
Yeah, I was trying to. The question is around the fact that, okay, cyclin A amplification, cyclin E amplification, sorry, is something that does happen in tumors, in human tumors. Have you looked at what's the correlation between cyclin E amplification and p53 status in tumor data, and how that fits with the model you're proposing here?
We haven't looked at it, but the cancer genomics studies that I showed in the presentation, I think they look at this, and they show that cyclin E amplification can induce whole genome doubling independently of p53 status. So yeah, I think this probably is the answer. Yeah, the point is, cyclin even if there is p53 positive, cyclin E expression can still drive cells to become whole genome double. I think this is what we are trying to say.
Okay, so there would be like two different mechanisms, let's say, something that you're describing in here, and something else that happens regardless of p53 being there or not.
Yeah, okay. Maybe. Yeah.
Okay.